All right. So we've had a really great introduction about all the utility of non-invasive genetics, different markers we can use. But I'm here to talk about how difficult non-invasive genetics can sometimes be as an approach, even given all of its really wonderful applications. So I have split up difficulties in non-invasive genetics into four main camps. The first one being low quality and low quantity DNA, representation of the actual genetic material, errors associated with data analysis, as well as all of the opportunities for contamination throughout the process. And I'm going to start with low quality and low quantity DNA because I think that that's where a lot of these difficulties actually stem from in the first place. So the DNA that we're actually looking at when we do non-invasive genetics that is of snow leopard origin actually comes from the epithelial cells of the snow leopard as the scat moves through its digestive tract. If we're doing something like diet analysis, then we're looking at the DNA of the prey items that the snow leopard consumed. So by the time the scat's deposited, it's already been through quite the journey. So the DNA itself has also gone through that as well. And once it's deposited, it's going to sit out in the elements and be exposed to temperature extremes, different weather conditions. And that means that the DNA is likely to degrade. So it's going to break apart when it's in those conditions. And so fresh scat, so the one in this picture, is really valuable. So me as a researcher, when I come across a fresh sample, I get really excited because that means the DNA is probably in better condition. But the reality is that when you are out sampling, you're more likely to find scat that looks something like this. So you can see this scat has been out in the elements for a period of time. It's starting to turn white. You can see that the outer casing, which is where we would actually find most of the snow leopard DNA, is kind of sloughing off there. And that doesn't necessarily mean that DNA is not valuable from that sample. It's just going to be more degraded and a little bit harder to work with. And degraded DNA is going to be a challenge that follows you throughout the process working in non-invasive genetics, from sample processing to data analysis itself. And when I say DNA degradation, I'm talking about the DNA breaking apart. So DNA is held together by a series of different bonds, and those bonds breaking apart result in degraded DNA. And so it's harder to work with DNA like this because you're kind of working with like different pieces of DNA instead of working with just a nice continuous double helix like you see in the top there. And so from the onset, no matter what you do as a scientist doing non-invasive genetics work, your DNA from the start is going to be low quality and low quantity no matter what you do. And so that's something to keep in mind as you're moving forward using non-invasive genetics. And it's one of the reasons why it's so important to use a preservation method immediately upon collection. So you'll hear us talk about, we'll get into this a little bit later in other modules as well, but one of the things that we tend to use is dry rite or silica desiccant. So when we collect, we're already putting the scat sample in something that's going to extract the moisture and help halt DNA degradation and halt things like nucleases from breaking down DNA any further. DNA extraction, once you have your actual sample and you get it to the lab, you've collected it, you're going to extract the DNA through a process called DNA extraction. And that's generally done with a commercial kit. Imogene showed a little bit of that. It's a series of different washing steps. And basically the result that you're going to get is the DNA that you're ultimately going to work with for your genotyping or whatever other analyses you're going to be doing with that DNA. And then extraction is a crucial step because DNA from scat in general has a lot of impurities and inhibitors attached to it. And so extraction is going to remove those impurities and inhibitors, which is going to make it easier on down the line for you to continue working with that DNA. Once you've extracted and have your DNA, you don't really know what you're working with in terms of quality and quantity until you measure that. And so we do that typically through a machine called NanoDrop. And what that's going to do is give us a score that's relevant to concentration in nanograms per microliter and give us a score of purity, which is a 260 over 280 score. So how pure is our DNA? And if you were to say GPS collar snow leopard and collect a blood sample, you would expect the concentration to be really high and the purity is probably going to be quite good as well. So this is just an example of a data set that I kind of randomly pulled from my own files just to give you an idea of the kind of concentration that we're looking for. Anything that I get basically that's in between 1.8 to 2, I would consider high quality DNA or good quality DNA. But you can see here on this chart that you're going to see numbers that kind of vary back and forth. And you're also going to see concentrations that range wildly. So you can see towards the middle there, I have one that's 110, And then towards the bottom, I've got a concentration of 0.8. So presumably having that lower quantity is going to be difficult to work with. And lower quality is also going to make that DNA ultimately more difficult to work with. But this is just to be expected because you're working with scat and you're not working with a tissue sample or a blood sample or something of that sort. One of the crucial steps after you're extracting and actually getting ready to work with your DNA is to amplify it. So the DNA as it is that you've extracted isn't enough to do analyses with. You're going to need to make repeated copies of it. And so we do that through a process called polymerase chain reaction, or PCR. So this is a thermocycler here in this background picture. And what that's going to do is, through a series of heating and cooling steps, make a ton of copies of our gene segment or our segments of interest out of the DNA that we're looking at. Unfortunately, with PCR, inhibitors that are in the DNA can really make that process difficult. So if you have any inhibitors that are left over from your DNA extraction, it's going to impact how well your gene segment that you're trying to make more copies of actually amplifies. And this is something to keep in mind when you're interpreting data. So for example, this is a species identification assay. We actually do this with cytochrome B, which was a gene that kind of got tossed out in Imogene's talk. It's mitochondrial. And there's a segment of cytochrome B that is specific to only snow leopards. So the first thing that we do when we actually are starting to work with our DNA is figure out, did this sample that I'm looking at, this DNA that I have, did it actually come from a snow leopard? On average, only about 30% of the scats we collect are actually snow leopard. There are a number of other species that it could be, other carnivores, things like Tibetan wolves, red foxes, even in some cases, domestic dogs, depending on what area of snow leopard habitat you're working in. And so what we do is we do the species identification assay. And if our genes actually amplify, then we would determine that to be a snow leopard. And amplification is denoted by these bars. So we use a process called gel electrophoresis to actually visualize our DNA. And so if we have a band here, this yellow color, then that would signify that our gene amplified and we have a snow leopard. And we do all of this in triplicate. So you'll see these different samples kind of repeated 3 times. And so if we got amplification, then we can say that that's a snow leopard, and that's great. But if it did not amplify, it could be one of 2 things, right? It could be that, yes, it's not really a snow leopard, or our PCR just didn't work. We didn't get amplification because our DNA quality or our quantity was too low, and we just had PCR failure altogether. And so those are things to actually take into consideration when you're actually talking about your results and reporting them with non-invasive genetics. And then that'll feed kind of into representation. And I'll just kind of briefly touch on this. But in general, because our DNA is degraded, we're going to have to use shorter sequences. So we're not going to be able to have this big long gene segment for us to look at, because that's likely not going to amplify with PCR, because we're just not going to have that nice intact stretch of DNA. So the shorter the sequences, the better likelihood we have of that sequence actually amplifying and us being able to work with it later successfully and actually get decent data out of it. But because we're using those shorter sequences, we have to use multiple markers to get enough genetic information that's representative of the sample. And one of the shorter markers that we typically use is microsatellites, which Imogene went into, which was great. It makes my job a little bit easier. So these are short tandem repeats. So they're just repeats of the same nucleotides over and over and over again. And they're relatively short, and they're variable between snow leopard individuals. And so what we'll do is that we'll amplify those to look at something like individual identification to build a genetic profile for each snow leopard. And then I'm going to go into combining data and analysis errors. So one of the main applications of non-invasive genetics is genotyping for individual identification. And just to break this down into a very kind of condensed nutshell here, if you're out in the field and you collect 4 scat samples, most of the time you're going to be collecting many more, but this is just a hypothetical. And then you construct 4 genetic profiles based on your microsatellites. Maybe you find that out of those 4, you have 2 unique genetic profiles determined, and that would tell you that you have 2 snow leopard individuals in that particular area where you're working. But the way that we actually analyze and get that information to build a genetic profile is via genotyping, is through something called an electrophorogram, which helps us visualize our actual microsatellites or our short tandem repeats. And so an electrophorogram is going to show us the length of the DNA fragments that we've amplified. And it's going to give us kind of an output that has these peaks here. So Imogene showed a few kind of little pictures of those, but essentially for every locus that we're going to look at, so this one, for example, is 894. That's at the top of each of those little graphs there. This is snow leopard SBC 109, again, running in triplicate. You're going to have peaks representative of the actual length of the fragment. So if there's 2 peaks, it would be a heterozygote, because remember, you have 2 copies of everything, one from mom, one from dad. If there's 1 peak, then it's going to be a homozygote. And so in this particular case, we have snow leopard SBC 109 is a heterozygote at 118 and 120 for this particular microsatellite locus 894. And we may change the number of microsatellites that we're looking at depending on the application. So for example, with individual identification, we usually do 7 to 8 microsatellites because that gives us a probability of identity that's sufficient. But if I'm looking at something like landscape genetics or something where I really want to like dig in a little bit deeper, then I'm going to use anywhere up to 32 microsatellites. And then this is just an example of a homozygote. So this is a different snow leopard individual. This is DUL123 at the same locus. And this is giving me a single peak at 110. So I would classify at this locus for this snow leopard to be a homozygote at 110. But these are all like really nice pictures. These are really nice electropherograms. Sometimes you're going to get ones that look like this, and it's because your DNA didn't amplify. And again, that goes back to having low quality and low quantity in PCR just not working. And so sometimes you'll have information that's completely useless because you can't define peaks. There's not nearly enough information for you to be able to actually make an allele call in this particular case for that particular sample, and then you lose that information. But it's just kind of the nature of the beast with working with scat DNA. And there are many sources of data analysis error or things that can kind of come in and mess with your data set. I'm going to briefly just touch on allele dropout because it's one of the most common in genotyping that we deal with. And that's when a heterozygote fails to amplify. And it's one of the reasons why, for example, it's so important to do things in multiple replicates. So for example, this is snow leopard SUJ12, and I have each of my 3 replicates here. And this is locus 1157. So this is the microsatellite that we're working with. And I would consider this snow leopard individual to be a heterozygote at 104 and 106. You can see in the 3rd replicate, I lost that 2nd peak. I completely lost the 106 allele altogether. So if I had only done, say, 1 replicate of this sample SUJ12 and I had allele dropout, I lost that allele, then I would call this snow leopard homozygote when in fact, if I had done it in more repetitions, I would have found that it's more likely and more accurate to say that that snow leopard is a heterozygote at 104 or 106. And then there's a slew of other things that happen, like amplification doesn't happen altogether. And then you can also have other things that occur that you need to be aware of when you're actually going through and scoring your alleles, and then ultimately taking that data when you're building your genetic profiles and doing analyses into consideration. And I'm going to touch on combining data because I feel like this is a really important one and something that's been kind of a beast to deal with in non-invasive genetics, specifically with microsatellites. And a lot of that comes down to allele calling and differences in allele calling, not to mention just differences in labs and how labs operate and human error and things of that nature. But a lot of it, I think, is due to positive controls. So for example, in our lab, we use the same 2 positive controls for everything. We know the genotype of those snow leopards forwards and backwards. We can be really confident that whatever allele calls are coming up for those positive controls, we can compare all of our experimental or the scat samples, the new DNA that we're looking at too. So for example, in this particular case, I have 3 different snow leopards here. They're all at the same locus at 347. And I have this peak here. I have a single peak. I would call this a homozygote. And I'm classifying it as 122, right? That's the length. And I'm comparing that back to my positive control at the same locus. So if my positive control at 347 has, for example, even numbers, right? So everything works in double, right? Because microsatellites. In this particular case, you can have tetranucleotides, which means there's 4 nucleotides, or dinucleotides, which means there's 2 nucleotides. This particular case is 2 nucleotides. So we would expect to only have every other kind of number as a peak. In this particular case, it's even. That's just what our positive control is. That's what we've called it. That's what we've done in our lab for years now. But someone else may have the same exact data set and call it 121 instead of 122 because their positive control uses odd numbers. Or maybe they found in a different region of snow leopard habitat where they sampled that allele sizes are much larger or have a greater range. So maybe they found an allele size all the way, for example, at 126. And nothing from our data set has ever gone to that size. And so in our particular allele bins, we don't have that number. So if you were to just kind of throw data sets together, it's not going to work. It's really important that they're standardized to positive controls, and there has to be open communication about what everybody's kind of doing. In some cases, best case scenario, you can, exchange positive controls and run them all in your own lab and do kind of that scenario where you're trying to match everything up, and that would allow you to combine data sets, but this inherently is very difficult to do. But if you had given the same data set to two different labs, you would likely have the same conservation outcomes or the same results, even though there might be a few differences based on allele scoring alone. And I'm going to talk about SNPs because SNPs are the future in this. We talk about microsatellites a lot. They're still a really great option. Microsatellites are something that's efficient to work with. There's a lot of literature about them. They're relatively inexpensive compared to doing sequencing with SNPs, for example. But SNPs are something that we're kind of gearing towards and moving towards as a discipline. And so, as Imogene said previously, SNPs are differences in just one nucleotide in one location of the genome from one snow leopard to the next. And what's really great about SNPs is that they're probably going to tell us more variation and adaptability of snow leopards than we would be able to infer from microsatellites themselves. Unfortunately, SNPs are really hard to use with SCAT. So you're actually looking for kind of these different, I like to think of it as different addresses on the genome, right? So we have a SNP panel and we're going to look at all these different locations on the genome, these different addresses. But actually targeting those addresses can be difficult because we're working with degraded DNA. So we're still working to develop protocols to actually make that a reality where we can work with SCAT and actually get good data, because we're still going to have issues similar to microsatellites where we're just losing information because the DNA is actually degraded. Our lab does some hypoxia research, and we look at specific genes and looking at differences in variation. So how are snow leopards in different elevations, how do the genes relative to how they can adapt to hypoxic conditions, are those different? And how does that relate to things like climate change or moving up in elevation or habitat shifts? And we've been trying to do that with SCAT, and it has been difficult. We've had some success, but even just looking at those specific genes and looking at SNPs has been quite the challenge. And so it is something that hopefully will get developed in the future, because it will be definitely changing for what we know about snow leopards, and it has particular application to combining data sets across institutions and making that a little bit easier. And then lastly, I'm going to touch on contamination risk. So contamination can happen at virtually every step with non-invasive genetics. Contamination in lab work and DNA in general is of concern. And I don't think a lot of people think about how every single step of this process, there's an opportunity for you to contaminate, and it's going to be hard to trace back. And once you have a contamination, it's going to follow you through the entire process. So it's important to be careful from the onset. So just for example, in the field, so this is a snow leopard latrine. So we've had multiple snow leopard individuals defecate in the same spot. And just those scats being in close proximity, there's greater likelihood that those scats could touch each other if the wind blows or it gets kicked by another animal. And so once those scats touch each other, then that DNA is on either scat from the opposing scat. So that's something to keep in mind and something to write down in your notes. During collection, you need to treat every scat sample as an independent sampling event. So you want to make sure that you're using a new collection tool every single time. You don't want to be repeating, for example, if you have gloves, you want to change a glove every time. If you're using tweezers, you need to change tweezers every time or sterilize them. So you want to make sure that you're not accidentally transferring DNA from one scat to the next scat that you collect. And when you're collecting 100 scats a day, this gets to be a little bit tedious. And sometimes when you're tired, it's important that you're keeping track of the steps while you're collecting to make sure you're practicing proper sample etiquette and collecting with the fresh collection tools as part of that. And then during transport and organization, so as you're actually putting scat in the tube, you're probably going to get some of that scat DNA on the outside of the tube. And then once all these tubes get kind of put together and organized for transport, that scat DNA is going to transfer to other tubes and so on and so forth. And so one of the things that we do when we first get samples in the lab is to take a 10% bleach solution and wipe down the outside of all the tubes just to try to get rid of, to kill any DNA, residual DNA that may be on the outside of the tubes, so that when we're working with them in the lab, we don't accidentally transfer with our own gloves. And then during extraction, scat, when dried, can be it puffs out. So as you're picking scat apart, you're actually going to have fecal material kind of like poofing out. And when you do that, you're obviously going to get DNA in different places of your lab space. So in the vicinity of where you're actually extracting. So you have to make sure that you're actually sterilizing in between every session, every sample that you're actually extracting. And then DNA aliquoting. So pipetting every once in a while, you might accidentally put one sample into another sample by accident, or you forgot to change your tip, or there are several avenues where during the actual process of pipetting, you can make some errors and accidentally contaminate. And so that's another thing to think about that's going to ultimately affect something like allele calling, because you're going to have all these crazy peaks because you have more than one snow leopard in your actual data set. And so in summary, I just want you to walk away from this knowing that non-invasive genetics has many challenges, but almost a lot of these are linked to low quality and low quantity. And if you're careful, they can be avoided. So you want to make sure that you're storing your samples properly, you're collecting them properly, you're doing DNA extraction to a sufficient degree, and this will ultimately reduce issues with PCR amplification, the way you score alleles and how accurate your allele scoring is, as well as data analysis down the line. Because our DNA is degraded, we have to use multiple shorter genetic markers to get enough genetic information. And different labs can have different positive controls and different ways of doing things in general. And so it is difficult to combine data sets, but it is possible. It just takes a lot of collaborative effort and can be something that is achieved. And then also contamination is a very real reality almost every step of the way. That's the nature of SCAT itself and the nature of lab work and working with DNA in general. So you have to be extremely careful about that as well. And that's all I have. So if there are any questions, if anyone wants to speak up or put them in the chat. I've seen the chat kind of like been bouncing around a little bit as I've been talking. So looks like there's some good conversation about standardizing and SNPs and microstats. And if anyone has any other questions.
Yeah, please, anyone, go ahead and any of the talks today, any general questions about this module, you know, about the things we've touched on. Yeah, we have plenty of time. So for any four of us to answer your questions. So Sherry, so yeah, if your video is not great or you're just too shy to speak up, feel free to type it in the chat room. So Sherry Young just asked, how long do we have between SCAT collection and actual analysis in the lab? So it's a great question with regards to timing, like how quickly should you process that sample once you collect it?
Yeah, so as fast as possible is the easy answer, but that's not always the reality. I think what's really important is for you to get preservative. So if you're collecting, make sure it's on silica desiccant. If that silica desiccant changes color, you need to replace the silica desiccant. Storage, you want to, you know, if you're in the field, moving from field site to field site, you want to make sure that you're trying to keep it at somewhat of a stable temperature. One of the benefits of where we work naturally with snow leopard habitat is that it's cold and dry. And so a lot of the SCAT samples that you're collecting are already in better shape than you would if you were doing research in the Amazon, for example. So you buy yourself a little bit of time there as well. But typically, if you have a SCAT sample that you can put into, for example, a -20 or -80 freezer, if you have access to that in your lab, or if you have a very consistent room temperature in your lab and you can keep them in silica and change out the silica desiccant when necessary, you can keep SCAT samples around for quite a bit. When I actually first started my PhD, Jan had samples from 10 years ago that I got DNA from and did analyses on, and it was fine. It probably would have been better. Maybe the allele calls would have been a little bit clearer if I had extracted it, you know, 10 years before that. But you want to do it as soon as possible. But if you've got the right storage, you can make DNA and your SCAT samples last quite a while.
Yeah, I want to just add to that. That's, you know, true. That's what we found, that it lasts a long time. I think the really critical thing is you want to make sure that the stuff stays dry and not humid, right? So if you have a place where that's you know, possible, then you know, you're good. But you definitely, if you're in a place where you're not sure about being able to keep it dry or things like that, then the best is just throw it in the freezer. And if it's frozen, then, you know, they're going to last for years. Yeah.
And just so in case it wasn't clear, there are different kinds of silica desiccant you can buy. It's worth investing in the color changing desiccant. So what happens is it changes color as it gets saturated with moisture. And if you see that your sample has changed, so it's often like an orange to white or purple to white or purple to blue, whatever. So if color change that you want to monitor, especially when you're in the field and as you get the samples to the lab, if you see a sample where the silica has all changed color, indicating it's saturated, is to change out that silica because it could mean there's still moisture in that sample. And if you're not, if you, for example, like, oh, I have a nice, dry environment to keep these in. If there's moisture still sealed up in that tube, you'll still get potential microbic activity to break down the DNA. So the color changing silica is probably a little more expensive than maybe just some regular cheap silica, but it's worth it so you can monitor the status of its action on your sample.
And it looks like we had a question about ethanol. What can be the best scat storing medium? Will ethanol affect scat quality? So I've seen both silica desiccant and ethanol used. Here's why I caution against ethanol from personal experience. So usually when you're collecting samples on your tube, you're going to write with a Sharpie marker, for example, what is in the tube. So you might mark a name, right, for that actual scat. Ethanol takes off Sharpie. So if your tubes leak, you're going to lose all that information on the outside of your tube. And it's happened to me before. We had some samples sent to us and they were beautiful. They were packaged great. The field team did an amazing job, but it had just leaked during transport. And so we lost, I couldn't match up the scat samples to the GPS coordinates because I didn't have the name of the scat sample to begin with. And evaporation as well as imaging says this can be a problem with ethanol.
On top of that, oh, sorry, go ahead, Jan.
I'm sorry to interrupt, but the other thing I would add is when you're trying to transport, especially on airplanes, right? Flying, across. I mean, if you're carrying hundreds of samples, right? You have 5 mils of ethanol in each one, then basically you're going to have potentially more volume of a flammable liquid than you can actually carry in your check-in. And then just every time you fly, it's just whenever you have something, a solution, the security and the people who are looking at it's they just don't like it. They don't like stuff in liquids because they're not sure what it is. So if that's why one of the best ways to do it really is just to try to keep it dry and transport the scat dry because you don't have to worry about transporting solutions. So now that's not always a problem in every single case, but that's an kind of added thing where in the lab we try to avoid using ethanol or any kind of liquids, especially going internationally. But ethanol definitely is a great preservative and it works if you're local. Other than the fact that, I also don't like using it for the reason Charlotte raised as far as like it can wash stuff off of the tubes. And just when you're in the field, liquids they're not that great for handling stuff in the field when it's cold and you got gloves on and things like that.
And then just to relate it back to the color changing, with the ethanol, you don't know when you've reached the saturation point with moisture. With the silica desiccants, with the color changing, you can say, okay, this needs to be changed out. You wouldn't know that with the ethanol. As far as I know, there's no color changing ethanol available to let you know when you've reached a saturation point with your water.
And I've had some samples sent to me that had been stored in ethanol. And so based on like some people in the chat are talking about different restrictions between transportation or between countries. In the past, we've had collaborators, like they will just, if the scat is stored in ethanol, they'll pour off the ethanol and ship it. And this only really applies if it can be shipped overnight and its temperature is not an issue. But because the sample had been stored in ethanol for a long period of time, in theory, the DNA was adequately preserved. And so they poured off the sample, sent the wet sample. And then when they arrived at their destination, you can either refill them up with ethanol or you can also put them on desiccant. And this only applies if the sample has been stored in ethanol for a long period of time, meaning that the DNA has been preserved. And if you are able to ship it in a temperature sensitive way, for as short of a duration of time as possible. I think the caveat here, though, is that ultimately there's a lot of ifs in all of those different steps. And so, yeah, the desiccant is probably still going to always be the best method. But I just wanted to add that in case you're working with anybody who has samples available and they're currently stored in ethanol, doesn't mean you can't use them. It just means you have to take care of that prior to being able to work with them.
Yeah, that's a good point. And remember when you do it the way that Imogene says, like once you if it's sitting in the ethanol, right? The ethanol kills all the bacteria, but probably most of the viruses. You pour that off, it dries, then it should be pretty stable. But like she mentioned that because there's an desiccant in there, you probably want to make sure it stays cold and it's pretty rapidly transported. So yeah, I also got to see. There's a email. Some countries have they definitely have restriction on transportation. So yeah, you really need to check shipping across countries, the rules and things like that. When you travel, if you can carry it like just in check-in or something, it's almost safer than trying to ship it. I found, and I think a lot of y'all had maybe had that experience. But you also, we need to start checking the regulations as well, because, for example, with the U.S., there's potentially constantly changing restrictions with the CDC, Fish and Wildlife Service, all this stuff. So just because we could have transported scat 5 or 10 years ago, we also don't want to give you the impression that it's possible that you can do it now. Even the other host countries, they're passing laws that restrict these things. And so if you're gonna, transport samples across boundaries, make sure you check currently what the laws and regulations are for those particular countries, because it's good not to make assumptions, because these things can change, by year. So just because we could do it in the past, make sure you double check that it's still possible in your particular case.
So there was a question from Vishnu asking what would be the best method of DNA extraction for old samples. So considering a QIAGEN stool kit or manual methods such as he cites the Boom et al. Or phenylchloroform methods, sort of we think across the variety of DNA extraction methods. If you have some old samples, what do you guys think would work best there?
Yeah, I mean, I would still go with the QIAGEN still kit method. I mean, that's the most we've had most experience with, and like Cheryl had mentioned, we've gotten stuff over 10 years old to come out well, or decently. But the phenylchloroform methods, they would work. They were actually prepared. Is that document up, the extraction document that we prepared?
I think it's on the GSLEP website.
Yeah, so maybe, is that possible to give them access to that?
Yeah, I'll provide that link. So I will send in the Google chat some of the PDFs that I mentioned in my talk, and Imogen and Charlotte can do the same for any of the PDFs they had. And I will also include a link to the four of us put together some protocols and recommendations on field collection and things like that. So I'll provide a link to those websites. I think it's on the PAWS GSLEP side. I could be wrong. Anyways, I'll track those down. They're out there somewhere. Send those along.
Yes, I guess to just direct it with the phenylchloroform, like because that kit was designed specifically for scat and stool samples, we tend to use that. And so I personally have not tried phenylchloroform on scat, but I mean, it probably would work. Your success rate might be a little lower. So it's like, if you can get the stool kit, get it. If for some reason you cannot, and you're limited to doing phenylchloroform extractions, then you could try. I would try it with like a set of 24 to 30 scats and see how that works. And the key with the scat extraction things is like, make sure that as you're doing these extractions, you monitor the results, you make sure that things are working well, because what you don't want to end up in is a situation where you extracted like 300 scats, you went back and your PCRs and nothing worked, So just kind of do it progressively and kind of double test the methods as you're working with them and as you go along.
So I'm just going through and sharing all the polling results and sort of getting a copy of those. And for people's own interest, just so you know who your fellow participants are, I will share those results. But it's encouraging to see lots of people considering using genetics moving forward. A fair number of you have some familiarity or comfort level with this information, and some don't, and that's great. Anyways, just so as you see them flashing by, I'm just going through and sharing these poll results. But please go ahead and keep asking questions.
I was going to add to what Jan was saying about with regards to DNA extraction for the QIAGEN kit. And so some of this information in more detail will be in the links that Byron is sending. But the QIAGEN mini kit is great because even though you have like a recipe that you follow in order to get your extraction, you can make little modifications to that kit based on what you expect to get for your DNA. So for example, we're going to expect out the gate that we're going to have reduced quality and quantity for DNA from scat samples because of all of the reasons that Charlotte identified. And certainly that can be the case even for a fresh scat. Although obviously that's not as much of a concern, that can still be an issue. And so certain modifications, whether it is adding an additional or adding an increased volume of proteinase K, or increasing the amount of cell lysis solution, or incubating your sample at a longer period of time at a slightly reduced temperature. All of these different small modifications can increase the efficacy of the individual process that step is working on in the kit, whether it's like lysing the cell or digesting proteins, et cetera. And you can also like further down for downstream analyses, when it comes to eluting your DNA, you can do second elutions in order to increase the concentration. And so, ultimately, all the things that Jan has said are true, but the benefit, I think, of these kits is as long as you have an understanding of what the flow through is doing in a given step, you can accelerate the process by simple modifications that don't really interrupt any kind of downstream analyses. And that'll be in more detail in the link that Byron's going to send.